Okay. Good afternoon, ladies and gentlemen. My name is Tina from Mindray headquarters. I'm responsible for the immuno product line, and today I'm taking around 15 minutes to share something new of Mindray Immunoassay new cardiac marker for high-sensitive TnI and NT-proBNP. So let's start the journey to explore more. I divided this topic into three parts. The first one is brief introduction of Mindray high-sensitive TnI and NT-proBNP technology. The second one is the performance of high-sensitive TnI, and the last one is the performance of Mindray NT-proBNP. Okay, let's go and look the first one. Actually, we are standing on the shoulder of giant high-test. High-test is one of the most famous raw material suppliers all over the world. I took this picture from high-test official website. This picture accurately summarized the high-test progression and development process and high-quality production in the fields of cardiomyopathy infarction prognosis, diagnosis, and heart failure diagnosis in the past 30 years. That is from 1994 to the present. Let's look at the achievement of high-test on cardiac, troponin, and NT-proBNP during the past 30 years. There are a lot of firsts here, but I would like to emphasize the highlighted ones. High-test is the first company to launch high-quality troponin monoclonal antibodies and to do researches on natriuretic peptides. And high-test is the first company to provide NIST international standard materials. And high-test proposed troponin and complex detection mode for the first time. And high-test, in the last year, high-test, a detection antibody for total NT-proBNP was developed by high-test. When we're talking about the whole target structure and antigen design of high-sensitive TnI, firstly, we should know the potential targeted molecules we could detect in the blood circulation in an MI patient. Actually, high-test found that over 90% TnI and TnIC and TnI are TnIC and TnITC. Actually, TnI is always bound to TnC, and almost no free TnI is found. So TnI appears in TnIC or TnITC complex. And to detect TnIC and TnITC complex can effectively avoid false negativity and improve the accuracy. And also, the IC monoclonal antibody design can improve IC sensitivity and specificity. When we're talking about the new technology on NT-proBNP, we benefit a lot from high-test. In response to the complex modification progress of NT-proBNP in vivo, high-test has developed a series of antibodies targeting different regions and forms of the full-segment antigen of NT-proBNP, using the complex of antibodies and antigens with the best affinity to ensure better clinical compliance. By analyzing the molecular structure and the target design of troponin and NT-proBNP, high-test developed high affinity, customized antibodies, and high specific antigens, which are integrated with Mindray reagent development technology to complete the development of a reagent and optimize high sensitivity and high specificity. Now, okay, well, how is the analytical and clinical performance of Mindray high TrPNI, actually there are three acceptance criteria for high sensitive TrPNI immunoassay. The first one is the CV at the 99 percentile level of the healthy population is less than 10, and the 99 percentile must be established according to gender. And the second one is the detection rate of the concentration between LOD and 99 percentile of the healthy population should be above 50%. And the last one is there should be 3 levels of quality control materials. And for total precision of Mindray high sensitive TrPNI is less than 5% at the 99 percentile level of the healthy population, which is stricter than the requirements of IFCC, AHA, and ESA recommendation dramatically, which was validated not only at Wuhan Asia Heart Hospital in China, but also at HHRI in the US. For the analytical sensitivity of Mindray high sensitive TrPNI, I listed in the table. The actual performance is much better than we announced in the IFU and better than that of our competitors as well. The overall detection rate in healthy population between LOD to 99 percentile, 99 to 90 is around 93%, 99% for males, and 87% for females, which meet the recommendation of AACC and IFCC guidelines. Furthermore, in the face of real scenarios, we conducted continuously researches in 6 hospitals covering 15 hospital departments, totally around 14,000 samples. Mindray high sensitive TrPNI shows very good anti-interference ability in routine samples with the percentage of erroneous results accounting for less than 0.01% of the total 14,000 samples. For the stability of Mindray high sensitive TrPNI, we monitored the stability during over 180 days, and no matter we changed the reagent kit lots and calibrator lots, the CV of the 3 quality controls were less than 5%, which shows Mindray high sensitive TrPNI had superior stability, which ensures to be used for no less than 180 days at customer site. Besides, we carried out some validations and evaluations both in China and in the US. This slide shows the evaluation in the US. The result has been exhibited on AACC 2023, which is conducted in Minneapolis, USA, to verify the analytical performance of Mindray high sensitive TrPNI immunoassay. And Professor Fred Apple, who is one of the most famous experts on heart disease field, gave a very positive comment on the analytical performance of Mindray high sensitive TrPNI. The other evaluation is at Wuhan Asia Heart Hospital in China. The scientific paper is available online now. The conclusion of the study is Mindray high sensitive TrPNI satisfies the definition of high sensitive TrPNI. It delivers excellent precision, sensitivity, and specificity, and it demonstrates excellent performance in clinical diagnosis, providing high levels of accuracy and safety. I also want to share some ongoing clinical studies with you. We are doing the research on 0-2 algorithm for rapid diagnosis, both in China and the US. Meanwhile, we are conducting clinical studies on risk stratification of cardiovascular events in special population. Moreover, we're trying to establish pediatric reference range for Mandarin high-sensitive troponin I in China. And in the following slides, I would like to share some preliminary results of some of these studies. In these slides, we did a study in Wuhan, Asia Heart Hospital with 950 samples. Mandarin high-sensitive troponin I performs the best when comparing diagnostic sensitivity, specificity, coincidence rate, PPV, and NPV with our competitors at 100%, 90%, 93%, 79%, and 100% respectively. Also, we compared the diagnostic efficacy of MI at a range of different concentrations, critical values. The diagnostic efficacy was maximized at 100 nanogram per liter with a clinical sensitivity of about 92%, clinical specificity about 98%, and a clinical coincidence rate of about 97%. And we have established adult reference range in China. Totally more than 8,000 samples were collected and tested in 2 hospitals in China to establish the 99th upper range limit for male and female. Here are the limits listed in the table. And the last one is about the establishment of pediatric reference range. We provide age-specific reference range limits for high-sensitive troponin I to our clients and recommend to validate for the local population. And the last one is a small summary. And high-sensitive troponin I is a true high-sensitive IC, and HiTest provided the raw materials and designed the reaction mode. And now the clinical cooperations, we have clinical cooperations with KOLs in China and abroad. And the last part is the analytical and the clinical performance of Mindray NT-proBNP. And when we're talking about the analytical performance, it usually refers to all of these in the slides. Here, I would like to emphasize how some of them are NT-proBNP repeatability, precision, interference, resistance, and stability. According to the guideline requirements on NT-proBNP, should have long-term stability and high precision. From the performance chart, we can see that all within run precision, between run precision and total precision with the series less than 5%, which satisfy the requirements of the guideline. Now, heart failure is a serious stage of cardiovascular disease involving a variety of organs and the complex medications. Heart failure patients on complex medications might have some interference on NT-proBNP test. So drug interference should be treated as a concern for NT-proBNP testing. From the left chart, we can see that Mindray NT-proBNP has good anti-interferences against the 55 medications. For the stability of Mindray high-sensitive NT-proBNP, no matter we change the reagent loads and calibrator loads, the series of quality control were less than 5%, which shows Mindray NT-proBNP has superior stability for no less than 180 days. And this is the master comparison between Mindray and Roche at different detection ranges. And it seems the results seem excellent, and the correlation coefficients are more than 0.989, close to 1, and the slopes are also close to 1. And for the clinical study of NT-proBNP, samples are collected for physical examination from a physical examination department for reference range establishment and validation. Other samples are for some clinical studies, including diagnosis, differential diagnosis of heart failure, pediatric monitoring of patients with heart failure, and also assessments of assessment of cardiac function in high-risk groups. And I would like to share with you some data and results of this study. We collected the samples from healthy people and healthy children for establishing the sex and age specific reference ranges and the pediatric reference ranges. After establishment, 22 samples from healthy people were collected for reference range validation. We only found one normal sample was tested to be positive, which is wrong. The overall positive rate was 0.45%, which is less than the acceptance criteria to 2.5%. That means the reference range are suitable. Ranges are suitable for the population, and and also we collected some samples to analyze and evaluated the clinical diagnostic efficacy based on the guideline recommended exclusion and the diagnostic threshold for the heart failure. And the data analysis showed the Mindray NT-proBNP has good diagnostic efficacy for both chronic heart failure and acute heart failure in the table. And the last one is a full summary. And actually, Mindray NT-proBNP is an ideal biomarker for heart failure. And the first thing is affinity. It has a good affinity and excellent accuracy. And also we provide age and sex specific reference range, and it has excellent anti-interference ability. And the last one is we have already done the adequate assessment. Okay, this is all for my presentation. Thank you so much. Thank you for listening.